This video presents the USGS U.S. Fish and Wildlife Bee Lab's standardized technique for processing bees at the Bee Lab. This was made as a supplement to the Handy Bee Manual and should not be used before contacting the Bee Lab or as a sole resource. Washing and drying collected bee specimens will result in better looking, easier to identify specimens. Pinning bees directly from water, glycol, or alcohol usually results in matted hairs and altered colors, along with a good coating of pollen, scales, and other detritus picked up from the sample. Many bee species are identified by hair characteristics that cannot be examined or properly identified if the hairs are matted or stuck together. The longer a specialist has to look at a poorly prepared specimen, the less time and funding they have in identifying additional bees. Washing and processing bees using the process described here will result in well-groomed specimens that exceed the quality found in unwashed specimens pinned directly from the field. Let's start at the beginning, with a container of collected specimens. The container will contain 0 to some specimens, as well as a filled-out tracking label. If the specimens were packaged into a Whirlpack or Ziploc bag, the bag will often be packed into a second bag, along with a paper towel to catch possible leaks. Several of these packages may be packaged together based on a shared piece of metadata, often location or date. These do not need to be kept together, but it is often useful to process everything contained in the Ziploc package during one processing session. The Bee Lab uses net bags made from mosquito netting to process large amounts of bees at once. Mosquito netting is a great vessel to process bees in because it allows water, dirt, and heat to move through the holes, while also preventing the smallest bees from escaping. The bag also keeps all the bees from a single container together, which makes for easy metadata tracking. The Bee Lab has developed a system to keep transect metadata linked with the specimens collected at that transect throughout the washing and drying process. The gem system consists of different combinations of colorful plastic gems glued to the inside of petri dishes. To link a container's metadata with its specimens, simply dump the loose copy of the gems glued in the petri dish into a net bag, then place a tracking label with the metadata into the petri dish. Double-check the loose copy of gems matches the glued gems before dumping into the net bag. If they do not match or there are missing gems, there is a box of extra gems that is kept by the processing station you can pull from. The specimens and metadata will be linked back together once the specimens have been washed and dried. Place the net bags into a tall cylindrical container with holes drilled in the bottom. At the Bee Lab, we use retired fiber powder containers and have found that they work well for us. The type of container used doesn't matter, as long as the net bags can be stretched over the opening and there are drainage holes at the bottom of the container. Pour in all the specimens from the container, in this case a Whirlpack, into the net bag. Remove any large plant pieces and soft-bodied insects, such as caterpillars, moths, and butterflies. You may also remove any larger arthropods that you know for a fact are not bees. Double-check that there are no smaller insects tucked away before discarding anything. At this point, the net bag should contain insect specimens and loose plastic gems. Make sure your net bag does not have any holes. If it does, set the net bag aside. If the bag is empty or does not contain any insects, write a label that says "No bees" and place that in a petri dish along with the tracking label. You do not need to do anything additional for this Whirlpack. Run the net bag in the cylindrical container under hot water for a few seconds. This initial rinse will remove the majority of loose dirt and small detritus off of the bees in preparation for their soap bath. Remove the net bag from the container and tie it shut. Draw the bag shut, then tie a knot into the bag to secure it in case the bag slides open in the dryer. Wrap the excess bag and string around the knot and knot again. Prepare a hot bath for the bees by half-filling a plastic container with hot water. Add in a couple squirts of Blue Dawn dish soap and swirl around so the water gets soapy. Dunk in your net bag and swish the bag around to wash the bees. Leave the bag in the water to soak while you repeat through the previous steps for a new set of specimens. Repeat the previous steps and prepare as many net bags as you want, or until your container fills. Once you have prepped your net bags, give all your bags another good swirl, then dump out the soapy water. Give the bags a rinse under hot water, then fill the plastic container again with hot water, but do not add soap this time. Add in the net bag to the bath again and give them another good swirl, then strain the bags out of the water. Pat the net bags dry using a towel. You can dry around 3 bags at once. The goal is to dry the bags enough so that they are not dripping water to speed up the drying process in the dryer. Repeat until you've dried off all your net bags. Gather up all your dried net bags and place them in the dryer. At the Bee Lab, we use a small RV dryer that has worked well for us. Make sure the knob is set to the highest setting, then close the door to start the dryer. Depending on the amount of bees you have washed, the drying process can take 15 to 30 minutes, with smaller bees taking less time to dry than larger, fluffier bees. Check the dryer every 5 to 10 minutes to see if the bees are done. You may remove bags that contain smaller bees and leave in bags with larger bees for longer if needed. Be careful when drying small bees, as they may fall into pieces if left in for too long. Once all your bees are dry, remove the bags from the dryer. Grab a piece of parchment paper and start by opening a net bag. Match the colored gems back to the corresponding petri dish containing a tracking label. Place the dried bees back into the petri dish along with their tracking label. If you will not be pinning the bees the same day, then close the petri dish and use an elastic band to secure the lid. As a reminder, this video presents the Bee Lab's technique for processing large amounts of bees. For more information on bee processing and links to resources, please refer to the Handy Bee Manual or any of the other resources below. We are working hard to create more useful videos for processing bees, but in the meantime, please refer to Sam Drogi's channel for more videos on bee processing techniques and other bee-related resources. Thank you for watching!